Okay, here we go. Well, thank you so much for joining us for the third day of our community workshop. This is the last day, but we have another 3 amazing working group today that I'm sure they will have interesting questions for all of you. So just a quick introduction. It's the same for the last 2 days, but for people that join us today, I want just to refresh what we have said yesterday and the day before. So this workshop is meant for providing a forum for the working group for presenting their work and ask questions to the AgBioData community regarding data sharing and archiving. So we really encourage everybody that use data, generate that data, and curate data to share their experience and thoughts. They are very important for us because we are going to implement your opinions in the following month and develop appropriate guidelines for fair data. So I want to remind everybody that we encourage a safe and productive environment that fosters open dialogue and exchange of scientific ideas. This environment is free of harassment and discrimination. We have a code of conduct. You can find it on our website. If you are experiencing or you notice harassment or misconduct today and you want to report it, you can send an email to agbiodata@gmail.com or to Susan McCarthy at smccarthy.agbiodata@gmail.com. Again, these contacts are available on our website, and so you can find them there. And with this, I want to say that
we'll also type in a question.
I want to say everybody to mute them yourself when you join. And also, so with this, I want to say that we really encourage everybody to share their opinion. And we try to be inclusive and respectful with everybody, regardless of their position. So we listen to understand, we don't interrupt, and we show empathy. And I want also to remind the host of the breakout room that it's also your responsibility to try to make people feel involved and also to try to make the environment very respectful. And so this is the general agenda. So this is the last day, but so today we will have 3 other working groups presenting with a presentation of 10 minutes each, and then there will be 5 minutes of a QA session after each talk. Then we will have a short break of 10 minutes, and then there will be the breakout room session, which will last 45 minutes. Each breakout room has a host, which is a member of the working group that will lead the discussion. This person will be also responsible to report at the end of the session. There is a scribe, which is a person that will be responsible of taking notes. I want just to remind the host and scribe, you are co-hosts of today's meeting. And so you can record the breakout room session, but please ask people if they want that. And you can also download on your local computer the chat of the breakout room session. And finally, there is a guardian, which is a member of the steering committee. This person is responsible for supervising the breakout room, that everything is going fine, and also is co-host of today's meeting, so will help me with the logistics and can also participate in the discussion. And finally, we will end with a short report for the working group, where they will present the main points of the discussion. And with this, I want to introduce today's working group. We will have the Ontology working group with Laurel Cooper, the Data Federation working group with Peter Selby, and the data collection with Olusola Ofuwabe. And so I want to remind everybody that if you have questions for the speakers or any suggestion, feel free to share them in the chat. I will be very happy to ask the question to the speaker at the end of their talk. And with this, I want to invite Laurel to share her screen and to start her presentation.
Yes, it helps if I unmute. Thank you very much, Anna Rita, for the introduction, and good morning, everyone, and happy St. Patrick's Day. So my name is Laurel Cooper. I'm co-chair of the AgBioData Ontology Working Group, and I'm here today representing the group. This talk was originally planned to be delivered by our main chair, Jennifer Woodward, but unfortunately she was not able to attend today, so I'm filling in for her. So what we want to do today is give a little progress report and update of what we've done so far in the last few months. My slides are not advancing. What's going on here? Hang on. Oh, there we go. Okay. So here's a quick overview of the presentation today. A little introduction of the working group and who's involved. A small introduction about ontologies, some progress that we've done to date. We, in the past 3 months, we've had about 7 meetings. We've reviewed a survey that was done in 2017 of the AgBioData member databases and what existing ontologies were being used and what existing resources people were using. And in the next steps, we're going to identify barriers to ontology use in the AgBioData community. And our goal is to provide recommendations and some decision-making tools to guide users, excuse me. And finally, we'll have some information about how you can get involved if you're interested. So the AgBioData working group is made up currently of about 19 members, and we have representatives who work on a variety of different areas. Strong representation from plants. We also have folks that work on animals, pest genomes. We have a couple of folks from the National Agriculture Library. And we have also some folks that work on soils, which is kind of interesting. I'm not sure how many people from the working group are here today, but we have a pretty wide representation. And of course, we're happy to have new members join if people are interested. So this slide gives a little generalized overview of the spectrum of different kinds of ontologies. Here, I'll move this off of the slide here. So ontologies are really like the most precise and complex type of control vocabularies that exist. This arrow spans the region of precision, semantic strength, and complexity, which also involves cost, of course. So ranging from simple glossaries, taxonomies, thesauri, and finally formal ontologies, which include things like relationships between the terms, axioms, and different restrictions in a hierarchy. So the use of these controlled ontologies of whatever, or control vocabularies, whatever type that you decide to use, can fit into the research workflow and can be an important component of the research workflow. And you can see this, we've identified this circle of the workflow where you could identify relevant data, aggregate it, and this involves, big data that a lot of us are working with now. Understanding and annotating this data with ontology terms is very important, and it allows us to analyze, interpret, and to gain more insights, which can lead to the deliverables that we all want to gain. So the aim of using ontologies is to enhance data search and discovery and to follow the FAIR principles, of course. So why would we want to use ontologies? What are the benefits? By using ontologies, you can bring together a lot of scientific connections. You can use the power of the formal logic in the ontology hierarchy to generate hypotheses. This allows you to do inferencing across scales, species, different disciplines, and vocabularies. This allows data integration across different granularities, vocabularies, and disciplines. Ontologies can help us discover data resources using the hierarchy of the ontology and different synonyms to expand your searches. And one of the powers of ontologies is knowledge representation in such things as a knowledge graph, where you can really ask a lot of complex queries, and this can be used to inform machine learning and natural language processing tools and methods. Excuse me for one sec. The Ontology Working Group objectives are to identify what resources are available and what possible gaps there may be. And our goal is to focus on use cases in the AgBioData community. So we're happy to have folks recommend use cases that they have, or maybe problems that they're dealing with, and how we can help you solve those. And in the long term, we want to provide some recommendations and tools that can be used to guide users. We did a little survey of what ontology resources are available at this time. And this is just a little list. And we can share these slides later, and then you can use these links if you want. Obo Foundry is an organization that is kind of a clearinghouse or a gathering place for a lot of ontologies. And they also provide a lot of standards for ontologies and metrics so that you can tell how good quality an ontology is. The EBI ontology lookup service is a very handy place to go and see a lot of different ontologies and connect to them. The CG centers have put together a list of ontologies for agriculture, and there's a list of them, the link to them there. It's another very handy tool. BioPortal and AgriPortal are ontology lookup sites as well. BioPortal is more broad ranging, whereas AgriPortal is more focused on ontologies for agriculture. And finally, Plantiome, the project that I work on with Pankaj Jaiswal, focuses on ontologies for plants. And we actually develop our own ontologies, the plant ontology, the trait ontology, and the plant experimental conditions ontology. And our partners over at Crop Ontology with Elizabeth Arnaud and Marie Angelique Laporte focus on specific standardized vocabularies and ontologies for specific crops. And I think they have around 40 different crops represented in the crop ontology. And we work closely with them at Plantiome. We have about 11 of their vocabularies integrated into the Plantiome as well. So there's a lot of resources out there, and I know it can be a little overwhelming for people sometimes to know what to use. So that's our goal, to sort of help people find their way through here. As I mentioned, there was a survey done in 2017 of the AgBioData database member databases. There was, this is just a screenshot from the white paper that was published in 2018 in database. There was about 29 respondents, and based on the different types of data that they work with, sequences, markers, QTL, germplasm, phenotypes, and genotypes, we had information on what people were using. This is the gene ontology and the sequence ontology. Those are very commonly used if you have sequence information. The plant ontology and the different types of trait ontologies are used more in like QTLs, germplasms, and phenotypes. And then the crop ontology is used in that same area too. And some folks actually have sort of an in-house ontology that they use, and there are some other reference ontologies used. So our goal is this year we want to update this information. So keep an eye open for a survey coming your way. And please let us know if there's any areas that, new areas that you'd like to see us focus on. So our plan for the deliverables for the ontology working group, as I mentioned, we're going to be sending out a survey of members and developing recommendations for the databases and the researchers. And we're thinking about maybe doing this in conjunction with the education working group. We talked with them yesterday about this. We're planning to host a webinar or maybe a hackathon where people can come in and do some hands-on training, working with ontology terms, doing some data annotation. And in the long run, we'd like to develop a white paper with our recommendations. So if you're interested in getting involved, the best thing to do is join the AgBioData community. And you can go to the AgBioData website. There's a link there where you can register, and that way you'll keep in touch with the different updates that are going on. Contact either Jennifer or myself at either of these emails, and you can also contact Anna Rita if you want to get involved, and she'll put you in touch with us. We have the AgBioData Slack is another place to communicate with folks, and we actually have an Ontologies working group channel, which we can invite you to once you're a member of the Slack. And we're happy to have you come to the Ontology working group meetings. Currently, we've done about 7 meetings, and I think we're going to get back to about a bi-weekly schedule, and we're going to try to do 2 different times a day through the month so that people from other parts of the world can attend at convenient times. So in the breakout room today, we're going to talk about what ontologies people are using and for what purposes, and what barriers you've experienced to using ontologies in your workflow, and what tools and recommendations you'd like to see us develop. And that's all I have. So I'm happy to take questions. Thank you.
Thank you, Laurel. for their presentation. I'm asking if someone has some question for Laurel. We have some minutes for that.
I can start out if if I may. Nice presentation, Laurel. Thanks.
Thank you.
And so there you have some more animal people who work on animal databases in your working group now, which might have been a change from the 2017 scenario. So have you identified, first, have you identified new ontologies to spread out to? I didn't have a chance to really scrutinize the slide that you showed that well to look for those, or is there a dearth there, do you expect? And also, do you expect that there'll be additional complications having a kind of a broader scope of the working group, or is one ontology for plant or animals the same as the other in terms of how the recommendations for how to use them?
Yeah, I think that's a really good question. And I forgot to mention, we actually have some folks that work on soils as well. And so that is two new areas, and it's not an area that I am an expert in for sure. So we'll definitely be exploring that. There's a number of model animal ontologies, such as mouse phenotype and different vertebrate trait ontologies and so on. But that is definitely an area we'd like to explore. And I'm really interested in exploring the soils side too, because that's an area that we really haven't explored too much. And maybe there's, a place there where people will want to develop some new ontology resources, but that's our goal, sort of to identify those gaps. I really appreciate your question, Monica. Yeah. And if there are folks, in the audience or in AgBioData who have other types of data that we're not seeing represented, we're happy to have you come and share what needs you have.
Well, thank you. I think Fiona McCarthy, she has a question for you, Laura.
Hi, and
Fiona.
thanks, Laurel. Hi, Laurel. Thanks for the talk. I had a question. You have this idea about doing the hackathons. Are you thinking in terms of people who have these large data sets to associate like metadata and information with it, or are you thinking more the granular literature annotation?
I think
or don't you know yet?
Yeah, at this point, we're really open to suggestions. But and so both of those ideas I think would be great. And maybe we have two different kinds. Yeah, I mean, we're really kind of going to be driven by what the community wants and what the community needs. So I think either of those, but I think having some hands-on experience makes it a little bit less intimidating for folks. And so, and then, we could pick out maybe a few. Those are both great use cases you suggested. So yeah, and I love your green today.
Thank you.
I think Doreen has a question. I see another hand up.
Yeah,
there.
so we will take another.
Okay.
Okay, Doreen.
Hi, and I'm not wearing green, I'm wearing red.
Oh, pinch.
Is there a reason why you're restricting the survey to AgBioData group and not extending it out so we bring in more people and advertise AgBioData a little more broadly, especially to animal groups?
That's a possibility. Yeah, I hadn't really thought about that because, we'd have to figure out, who to email and who to contact in those areas. But if there are people who have, might be interested, we'd be happy to include them. There's no reason we wouldn't want to. It's just, under the umbrella of AgBioData, that was our thinking, that we would send it out to all the, the members or the participants. But of course, there are people who have not joined AgBioData yet who would love to bring in and expand, maybe work with the recruiting working group in that area. It's a great idea.
Yeah, we'll have this. Okay, thanks.
Yeah, it's a great idea.
Yeah, you can reach out to them, but also if you need any help, I can also help you with that. Okay, thanks, Laurel, for the great presentation. I think if there are other comments in the chat, so feel free to follow that. And with this, I want to invite Peter from the Data Federation Working Group to share the screen and present.
Let's see if this actually works. Oh, it was working just a second ago. There it goes.
Yeah.
Okay.
Okay, great.
Yeah, so hi everyone. My name's Peter Selby. I'm the BRAPI project coordinator. I'm based here at Cornell University, and I'm presenting on behalf of the Data Federation Working Group today. We've sort of gone with a unique title for our presentation. What the heck do you want your database to say? We'll explore to why we have that title a little bit later. So first, we'll introduce the Data Federation Working Group. Chair Jennifer Clark and co-chair Andrew Farmer. We have a somewhat smaller group, and sort of echoing what a lot of people have said this week, always welcome new members. This working group is a little bit more technically focused, but that shouldn't stop you from joining. Sometimes we get the best insights from explaining some of these technical ideas and realizing we don't all agree on the same definitions of everything. So yeah, please welcoming new members to explore some of these more technical concepts. So yeah, I think the first challenge we were faced with is trying to define what this working group was and sort of what our target goals should be. And so we really tried to define sort of data federation and sort of where it fits into what we can reasonably achieve. So we have sort of these formal definitions for data sharing, just being able to share data between tools and between people. Programmatic data access, being able to access data programmatically using code and making data computer readable. And then sort of the data federation is sort of the highest, sort of most lofty level, where the collection of databases within the federation are all highly interoperable and sort of appear to be a single system to the outside world. And to try and sort of wrap our heads around that, we've sort of had the data sharing spectrum. And so everything on this slide counts as data sharing. If you have physical documents that you're going by snail mail, sending back and forth by snail mail, that counts as data sharing. But we're just sort of looking on the spectrum of automation and sort of the less automation versus the more automation. So once we sort of get into more automation, we get into programmatic data access, looking at APIs and accessing data programmatically through software. And once we get into sort of the highest level of that, data federation, where all these systems are highly interoperable and sharing data back and forth, maybe without the user even knowing that data sharing is happening. So trying to wrap our heads around the different sort of at the top, the different technologies or protocols for sharing this data, and along the bottom, the different styles and data structures that this data can take as it moves between different systems. The other thing we're trying to sort of define is the interoperability and how that factors into everything. So looking at, and I'll say here also, we have a huge overlap with some of the other working groups, and we'd like to try and collaborate with them a little bit more. So we're talking about semantic interoperability. That's the sort of definitions of data and terms. We just heard about ontologies from Laurel. and some of the things in the genome nomenclature yesterday. Sort of making sure that things are well defined. Looking at syntactic interoperability. This is the structure of the data, how things come together. Again, the different working groups, the genome to phenome, the standards for genetic variation working groups, all these different groups looking at standards of the structure of the different data. And I've sort of put a little sort of contrived example here in the definition, just as an example of data structure, to call out really the difference between human readable data structure and computer data structure, and the fact that we can see a human could read the taxon example here. We could read both these taxon examples and interpret that very quickly, but a computer would struggle even with this just slight variation of sort of the reverse of terms. The computer would struggle to interpret that properly. So having more rigid structures and syntax for interoperability is really important. And sort of protocol, the highest level protocol interoperability is the mechanism, and we saw some of those before. Having similar mechanisms and protocols for actually moving data from one system to another is really important. Yeah, so you could have two databases that have exactly the same semantics and exactly the same syntax, but if one of them is transferring data by file download and the other is transferring by API, they're not going to be very interoperable, just because they're not using the same protocol. So yeah, looking at sort of the breadth of this problem in terms of data federation and data sharing, we've got the different types of interoperability we need to look at, and this huge spectrum of data sharing that we need to look at. And the working group was somewhat concerned about sort of this this effort. It's sort of a large effort, and I shared this story from my work with the BRAPI project. I was hearing a lot of input from stakeholders saying, I want all my tools to be able to talk to each other. I want fully integrated systems and fully interoperable systems, and we need complete data federation. And so I said, okay, I will snap my fingers. All of your systems can talk to each other. All the technical challenges are solved. What the heck do you want your database to actually say? Oh, that was supposed to be a big reveal of the slide and it's not moving. Dang it. Yeah, so this is the question, is what are the actual use cases? What pieces of data do you want moved from system to system? What is actually useful? Why do you want data federation? So that's really where we are now as a working group. We're really trying to dig into what are the concrete use cases that we can focus on as a group? What specific types of data need to be shared? And that will drive sort of the technical solutions. What level of data sharing do we need? What technical solutions do we need? So yeah, so our group goals for year one, similar to the Ontology group, we had a previous survey that was done in 2018 that we've been looking at. So we want to send out another one, reassess the community's data sharing needs, and yeah, so we can compare, see what has changed over the past 4 years in terms of technology and data sharing, and try to see where we can go from there. So keep an eye out for that. Once we start collecting some of those use cases, we want to try and define some of the repeated use cases, some of the common or really important data share needs within the AgBioData community, so that we can focus on those areas and really try to address the needs of the community. And then sort of some sort of white paper or set of use cases that can be passed on to next year's working group to really try and start addressing these technical challenges. So yeah, for our collective discussion later, we really have one big question, which is, what are your use cases? And that's going to be totally the focus of our discussion, is what are all the different types of data sharing use cases you'd like to see? What are the important ones? You know, what should we prioritize? And we've got some helper questions here to try and flesh out these use cases, get some ideas flowing. And I do have, there's some example use cases that we've put together, but we can dive into those a little bit more in the discussion to have some ideas to focus on. So with that, yeah, I'll end and take any questions.
Thanks, Peter. So there is one question in the chat that I'm going to read out. So Timothy is asking, where do you see bulk data files on an FTP access in your spectrum of automation?
So that, I mean, it depends a little bit on the structure of the files, but you know, that can be accessed programmatically. So I think it would fall into programmatic access, but it would not be, it would be difficult for it to be ready for data federation. So yeah, well, it depends on the structures of the files and what their actual contents is, how interoperable that data is. But generally, from an automation standpoint, FTP is okay to automate. It can be done, but it's not the easiest way to do it. So.
Thank you. I have a question, like, I don't know if it's related, but you know, I guess there are new type of data that are like having breeders or geneticists, which are like, you know, high throughput phenotypic data. And there are sometimes you have very big, very large data that sometimes there are no place where to put them. I imagine, let's say, drone data, they are so big that some people, there are not even repository yet for high throughput phenotypic data. So I'm just asking, like, is the size of the data also an issue that also kind of limit data sharing and also the interoperability between databases?
Certainly for the efficiency of things, it's in a lot of cases, if you have a proper data repository for these large data sets, they can be made interoperable with the rest of the systems, just pretty in standard ways. I would say that the size really comes down to efficiency, and one of the things that you can look at doing if you've really got large data sets is instead of moving the data from system to system, you can move the tools closer to the data, and that requires its own level of standardization. So you can say, you know, this is a huge data set, but it follows these standard APIs and interfaces. So we can take this tool from a different system designed for smaller data sets and put it, you know, bring it closer onto the same machine as this large data set. So there are ways around getting around those efficiency problems with large data. But yes, it is another technical concern.
Yeah. Well, let's take the last question from Rex Nelson.
Hi. In regards to the large data, such as the spectral data, I think there's two issues here. One is the original data and one is the analyzed data. If we are to believe anybody's paper, the analyzed data should be sufficient for everyone, and that would really reduce the size of the files if you could say strain X had value Y.
Mm-hmm. Yeah, you know, and those are the type of use cases and information the working group needs, right? You know, we need to know, do we need to look at solutions that can handle the large original data sets? Is that something that people will often be looking at? Or is it enough to be looking at the analysis results and looking at those smaller data sets? You know, what exactly do we need to focus on? What will get the most value out of the data in terms of solutions for data sharing?
Okay, thanks, Peter. Thank you for the presentation. And I guess if you guys are interested, please join the breakout room of the Data Federation. Now we will move on with the last presentation from the data collection working group with Sola. So I will share the screen for him. Yes, Sola, so if you want, I want also to remind everybody to mute yourself during the presentation. Thank you. When you want, Sola.
Yes, thank you, Anarita. Welcome, everyone. My name is Shola. I am making this presentation on behalf of data collection, accuracy, and precision. And we'll be looking at the role of the working group as concerning reproducible research. Please, next slide, please. Yes, the mission of the working group is for us to double check data and ensure that the data are of high standard, data that are free from errors. Next slide. Yes, the working group schedule is we meet on Tuesdays fortnightly, and the time is between 3 and 4 p.m. GMT +1. We encourage new members to join the group. If you are interested in being part of our group, you can contact Akim or you can contact me. That's our email address on the screen. Next slide. Yes, the present challenge of the working group is lack of commitment by some of the members. We don't have much representation, so we highly encourage others to be part of our working group. Next slide. Yeah, what is data collection, accuracy, and precision? I try to make this simple. Collection means to collate data and metadata from different sources. This is about to the accepted standard. Accuracy talk about the level of agreement. Then precision is about repeatability of a measurement. Precision illustrates the level of variability amongst the value you collected. Next slide. Thank you. Now, this is just a pictorial illustration. The focus of the working group is to ensure that the data we have is of high accuracy and high precision. The next slide. Now, I try to make a little explanation. Data curation. Data curation is about organization and integration of data from multiple sources, the enhancements of this data and maintenance over time, and also active management of research data to produce, the next slide, findable, accessible, interoperable, and reusable data set. The next slide. Yes, this is just the life cycle of data curation. We have a research question, you seek for the data, you manage the data, then you do some analysis with the data, then afterwards you can archive the data for future use, then you make a publication. The next slide. Now, the relevance of data curation is an avenue for data professionals and researchers to collaborate. Data curation ensures the validation of statistical claims in research. Also, you can't talk about data curation without talking, you can't talk about reproducibility without talking about data curation. And the data curation is also the origin of software curation, because you require softwares to be able to effectively engage in data curation. The next slide. These are some of the tools that you can use in a curation and reproducibility. We have Python, we have Power Studio, we have, and the other ones, please. The next slide, please. Now, the task of the current task of the working group is to review the challenges associated with acquisition of phenotypic data, with focus on the tools for current tools used in phenotypic data and the metadata collection, the method of collection of phenotypic data, and the phenotypic data analysis, interpretation, and reporting. The next slide. Some of the findings that we were able to come up with, some of the tools like satellite imaging, tools used for satellite imaging, some tools like handheld sensors for estimation of chlorophyll fluorescence, tools like, so this kind of that by the weather, depending on the weather. Also, these tools can also be affected by the frequency of imaging. It is highly high resolution image is preferred to low resolution. Also, some of these tools are expensive. They could quite be expensive. Then the time to process some of the image acquired through this tool is a bit long. Another tool used in phenotypic data collection is unmanned aerial vehicles like parachutes, like rotocopters. Yes, these two are very effective, but they can produce some auto mosaics that will result in data inaccuracies, lens distortion, ground sample distance, image overlap are some of the factors that contribute to auto mosaic production. Also, as mentioned earlier, softwares, you can't separate software from data curation from phenotypic data collection. So some of the software used for phenotypic data, we have some of them are listed. FenStat, that's an R package. Then we have Bioconductor, and that's, I mean, Python for those using Python for analysis. Yes, these tools are very essential and very effective, but the entrance is you need to have the knowledge of R, or you have to have the knowledge of Python or the knowledge of this tool to be able to effectively utilize this tool for phenotypic data collection. Then most of after the collection, you will need to analyze the data. Now, versatility in advanced mathematical and statistical methods are also essential for effective analysis and correlation. In humans, there is what is called digital phenotyping. And I try to define it as, it was defined as an in situ moment-to-moment quantification of phenotype with the aid of personal digital devices like smartphone. Yes, you can use your smartphone to acquire some digital phenotypic data from human beings. But the problem is, the issue with this is the issue of privacy and the issue of data security. Those are some of the hindrances that can impede the effective collection of digital phenotypic data from humans. Yes, the next slide. Yes, just try to highlight the process of acquisition of data and plant phenotyping and data accumulation. You get your samples, then you use the image acquisition devices like satellite sensors, then you process the data with some of the software earlier mentioned, then you accumulate the data. Then molecular data, environmental data, the next slide. Yes, I try to show the workflow for image analysis. For most of these data, a lot of them require imaging. So when you acquire the data, you use software like EBimage. That's another package to analyze some of this data. The next slide. Yes, depending on the type of analysis you want to do, we have phenotyping for breeding and phenotyping for precision. There's just a little difference between that. Phenotyping for this precision is about management, how you manage the farm, but phenotyping for precision is about how well you breed your plants or you breed your animals. So there is just a little variation between them. So you have to put that into consideration when you are trying to acquire the data for the research.
So that you have like 1-2 minutes left.
Okay. Now, some of the challenges we are having for phenotypic data sharing is as listed in the slide. Then these are the members of the working group. Yes, that's the last slide. Thank you very much.
Thanks, Sola. So thanks, Sola, for your presentation. I think your working group is very important because it kind of connects with all the other. So there is one question from Monica. These are great points, Sola. Have you looked into the data carpentry initiative? It may be worth looking into their recommendation, in particular for smaller custom data set and analysis.
Okay, we'll put that into consideration. Data carpentry.
Yeah, you can see that also. I can also share with you the link to data carpentries, and I guess this is will also be good to discuss during the breakout room session.
Okay.
The other thing that I see here is that the data collection working group really connects with many other working groups, like let's say genotype to phenotype, and also like, you know, Deontology. So did you guys reach out to any of the other working groups? So let's say you can also have joint meetings to see if there are overlapping points that you can also work together.
No.
Yeah, so maybe, yeah.
Because
So, go ahead, sorry.
I should go ahead.
Yeah, so there is one question also
Yes.
for Leonard. This is the last question, then we will go very quickly to the break. Also, with respect to specifics of what data needs to be tracked, have you looked into the MIAPE standards? I don't know how to say that. But yeah, so do you know about that? You can read about the questions in the chat if you want.
Okay.
Minimum information. Yes.
I didn't get the question.
Okay, yeah, so thank you, Sola. I think we can move with the, so now we will start very quickly. We will have a short break, and then we will move on with the breakout rooms. Before we start, I want to say that the breakout room are now I'm going to open now the breakout room. And so today we have 3. If you are not very familiar with how to join the breakout room, there is, so you can click on the sign below your window, and then you will have this window popping up, and you can join any of them. If you need any help, please let me know. I can also help you joining any of them. So the breakout room will last 45 minutes. And I will send a reminder that I'm closing the room 1 minute before. So please feel free to join the room. They are open now, but we will officially start the discussion in 6 minutes, so 11 a.m. Eastern time. Hi everybody, welcome back. I hope you guys had a great discussion during the breakout room. So now it's the time for the host to report about what you discussed during the breakout room. So the first group reporting is Deontology. So I don't know if Laurel or Clement will be.
Hello again. We are both there. Look, we can do that with 2 voices. I mean, Laurel was there.
You go ahead,
and I can stop maybe. And so just to give you a little, how long do we have for that? Like 5 minutes?
Yeah. So if you want, you can also share your screen if there is anything to show. Otherwise, you are free to.
I told Laurel why she should be able to share the screen. She's been doing that all the meetings. Maybe you can show the notes, Laurel, while I'm talking.
You can also share now because I make you also co-host, so you can.
Okay, yeah, I can do that here also. Google Chrome here, it's okay. No, easy. So overall, so you should be able to see the notes we have for our meetings. But we had 3 main little aspects that we wanted to mention. I just recall them here, and then I said a few words about them. What ontologies are currently being used and for what purposes? That was our first asked question. What are the barriers to using ontologies in your workflow? So having some feedback and then what tools and recommendations are needed. So we spent a lot of moment on the first aspect in order to refresh some of the review and heard about the ontologies that are being used by people in the group in the breakout. I did refer to some kind of similar review that was done a few years ago, and that was captured. And so we had a list of all the ontologies that were used by AgBioData database, and that would be interesting to update that. We will need to go behind such breakout session to actually finalize such an update, but we heard about some of the classic ontologies that are important in the field, like gene ontology, plant ontology, crop ontology, the agronomy ontology that is also now more recent than the review we had a few years ago on that. So we definitively see that among, we haven't finished the tour in the group there, but we see that there are some reference ontologies that are being produced by important groups and that are still like playing the role of the pivot and things like that, because they are showing up in different databases. So that was kind of one of the lessons learned on that. We also kind of tried to evaluate if there were some different levels of semantics being used by different groups, not necessarily ontology at the top of the spectrum in the more formal level of semantic use, but also standard thesauri or standard vocabularies or things that could be used like AgBioData for indexing data and things like that. AgBioData was mentioned at least once, I think, during the tour. Anything that we should say else here? What do you think about them? Then, okay, so definitively hearing about the different ontologies being used by each group is still, so I was going back to the program here, is still quite important, at least to hear about things and maybe new resources. Then we start talking, I mean, in parallel, we kind of start talking about some aspect in using ontologies in your workflow. So we spend quite an important moment on the whole process that is very important to different communities when they are maintaining ontologies and developing ontologies. Ontologies are alive. They are living objects, and a lot of communities have moved to things like GitHub to actually provide feedback on their ontology or get issues from users to submit new terms, submit new threads, things like that. And so we discuss what are the blocking steps or the blocking aspect in using GitHub. Are there some training needed? Is that convenient or not? How do we connect the place where the ontology is released or the ontology is browsed, and then and the feedback is given also to the user. So that raised some question there. We also identified in this part of the discussion, I would say, we also identified the fact that ontologies were also coming from the scientific domains, and where the vocabulary that the scientists use, or let's say the academic domain would use, and sometimes the connection with the day-to-day language that will be used by the farmers or by the farming industry or other actors is also necessary, and they might not necessarily mean the same thing. And so there is a bridge that needs to be created there. That point was also discussed several times during the breakout, and new stuff also being done in the domain of agriculture, like food ontologies or nutrition ontologies that are like more recent domain areas that are covered by ontologies were also pointed out. And it's important probably also for our database to also maybe complete their spectrum also by also going down to the end of the line and understanding what kind of food can be done with the different models and crop that are being studied in our database and things like that. And then finally, and I'll wrap it up on that, we didn't really have time to talk about tools and recommended things. So I kindly ask everyone to put some stuff, and you see everyone playing the game right now. We have the tools and tools. So I catch a sentence that was important, which is we have tools for editing and annotating. I think it's important. I think Tanya, you say that at the end. So there have been things being done in the ontology world for editing and using those ontology for annotating, but I catch also the need for being able to select and recommend the ontologies to use. And so I've been doing some work personal on that in the past, but I know there is still a gap in helping people to identify which ontologies to use and how to select them. That's also an important aspect. So I think as also a provider of services on ontologies, that's also words that we can hear that we need to also continue improving the mechanism for selecting and recommending ontologies. And so if I had personally your time to say something in that session, that part of the I would have said that we are working also on making ontology fair object and evaluating the level of fairness. And I don't know if any other topics in the groups, in any other breakouts, I've mentioned the notion of fair and things like that, but this is something that we are currently doing also to facilitate the work of recommending and selecting ontologies. And so, yeah, I think that's more or less the idea of what the things that we're discussing in the group.
Well, thank you, Clement, for sharing this. I guess you guys will still be working for the next month on this. Yeah, so now it's time for the Data Federation. I guess Andrew is going to report about the Data Federation Working Group. Yes, great. Andrew, are you muted, or guess you're muted because I cannot hear you. Oh, I think you can. We cannot hear you, Andrew. Do you guys can hear him? No.
No, I can't.
Okay, so it's not my problem. It's about, okay, because I was worrying. Okay, so maybe we can go on with the data collection while Andrew is trying to figure out, or otherwise, I don't know if anybody else from the data.
Can you hear me now?
Yeah, we can.
Oh.
So yeah.
Okay. Sorry about that. I must have jostled my connection. All right. Well, I won't try to screen share my notes, but I'll just quickly summarize. So we structured our discussion around use cases, and I think there were kind of three main things, I guess, that bubbled up to the surface for me in terms of what we discussed. One is that most of the data resources are fairly entrenched in the technology stacks that they have chosen. And so, you know, perhaps one thing that we need to be thinking about is how to take existing systems and put layers on top of them that facilitate exchange of data rather than necessarily, you know, trying to design new systems that would have that baked into them, because a lot of the sites that we're thinking about are fairly resource constrained and wouldn't easily be able to just pick up a new system and implement it at their site. Another major topic that came up for us was, you know, as we start to collect data across multiple systems, there's going to be a lot of heterogeneity in terms of the standards that people have used to apply to their collection of data. And, you know, how do you give that information to the user in such a way that they can make kind of informed choices? So, for example, you know, is there experimental evidence associated with the data from, you know, these gene associations in TER, maybe everything from soy basis computational, and certainly there are things like Go evidence codes that capture some of that. But we also talked about use cases where, you know, the judgment that would need to be made would be more complicated. For example, you know, when comparing genomes and trying to make inferences about copy number variation or presence absence variation, you really do need complex kind of data quality metrics of the sort that the genome nomenclature working group kind of talked about in terms of BUSCO scores or things like that, to be able to really evaluate, well, am I not seeing a gene, you know, because it's really not there, or am I not seeing it because this particular data set, you know, just is likely to not have it represented for some reason. And the third thing that we talked about, well, I guess as part of that too, we talked a little bit about kind of the implications for super automated data processing, like machine learning methods, and, you know, how
how the variability among data being brought together might be solved, or pose challenges to those types of methods. And then finally, just in terms of kind of how we go about really thinking about what the use cases are that are of interest to the community, there was a little bit of discussion of the notion of a kind of a Stack Overflow for our databases, where we encourage people to say, well, this is the sort of question I really want to be able to ask of the databases in general. There, maybe there is an existing way to ask the question and somebody can just answer it, but just being able to kind of have a sense for what people want to do and what the rankings associated with those types of questions, how many other people in the community might be having those questions, crowdsourcing those sorts of operations rather than having somebody, a small group of people trying to decide, well, what are the relevant use cases, could be very powerful. And I think that is a pretty good summary of what we talked about, but the notes will be available. Thanks.
So much. Now the last report is for the data collection. Akim, if you want to shortly report about it, thank you.
Akim, I think you're muted.
Yeah, I'm gonna ask him to unmute. Okay. Now.
Thank you.
No, thank you.
So in the break room session, we discussed basically about issues and challenges associated with data collection. We basically asked 6 questions, and the questions borders around what are the challenges associated with data collection, accuracy of the collection, precision of the analysis, accessibility, and fair sharing. So the responses are as follows. 1, it was unanimously agreed that there is no set of standards for data collection, and hence there are ambiguities associated with data collection and accuracy. So now we said, okay, since there are no standards for collection of data, in order to overcome the challenge of inaccuracy and lack of precision, researchers need to curate their data collected in an accessible repositories, either at their lab level or institution-based level, to enable reusability of the data and correctness. Probably someone may raise a flag that this data was not appropriately corrected or curated. We also identified that data should be used and shared. They should not be discarded. We should not say, oh, this data is not correct, and as a result of that, we are discarding this data. No. We should make sure that any data collected is used, because this will promote data sharing access, and it can also reduce the cost with the generation and sharing of data. So in every data collection, we need to ensure that there is information about where and whom collected the data and what are the barriers for accessing this data, so that another person planning to do such things will know what he or she should be expecting. Then we identify the need for public-private partnership in data generation because of some confounding issues associated with collection of data, sharing of data, and accessibility of data. So if there is public-private partnership, there could be resources that can enhance collection of data and accessibility of the data. We should also make sure that when we are collecting data, we consider geopolitical consideration of dispersing data collection, so that whatever data we are collecting for a particular area will be representing true point of that area. So we also identify that data storage and good standard of data formatting and usage are necessary. So we need to consider computer readability versus human readability. So because this could be important in storing the data and the formatting of the data for future usage. Since data collection is a wide issue and the accuracy and precision are not standardized, then there is need to narrow the purpose of data collection to form part of the research design. This will be critical and it will assist in the fair usage and free usage of the data. Then collection of phenotypic data of animals by collaborating with indigenous community, as well as educating the indigenous community population is important, because if we are to collect phenotypic data from a community of indigenous people, if we have education plan for them, they may see that as a benefit, and as a result, they will cooperate with us in gathering the data. And also, Sunita suggested that the use of data mining tools may be a way to affect inaccessibility of databases. Now, while we consider all those responses, we made these three important recommendations. That since there are no set of standard for data collection, now the question is that, can we establish a standard for collection of phenotypic trait data so that it will narrow it to that level? We may be getting it right from there. Then while working with indigenous community for collection of phenotypic data, there is need to include educating the indigenous population so that they will see it as being part of the project. And the last thing is that it is important to engage reviewers and editor of journals in deciding on standard for data collection, so that if anyone is reporting a method for collection of data in their manuscripts, the reviewers and editor will be aware, and they may not be able to raise a flag against the approach or the procedures. So those are issues we discussed. Thank you very much.
Thank you so much, Akim, for sharing your notes. And with this, I want to just thank you, all of the speakers and all the people that joined us today and the last 2 days. You guys did a great job, and I hope all the feedback that the working group received, they will be helpful for their work. I just want to remind everybody, so the working group members, but also all the community that joined for our community workshop, that the working group will report about how they are going to implement the feedback that they receive during these 3 days in the next monthly AgBioData webinar on April 6th. So please stay tuned and we will share more information about it. And with this, I want to again to say thank you to everybody. And if you haven't done yet, you can join us by becoming a member. Please visit our website. We also have a Slack channel. It is a very good place where we share all resources and information. And also it is a good place to contact and reach out for the working group. We have a Twitter account where we also post events. If you need any assistance or if you are interested in joining any of the working group, please send an email to agbiodata@gmail.com, and we are also on LinkedIn. And with this, I want to thank you, all of you, for joining us, and thank you so much. We hope to see you on April 6th. Thank you.